Hello and welcome to this Technology Networks webinar, Reviving the Past, Safeguarding the Future, Challenges and Innovations in Cell Culture for De-Extinction. I'm Dr. Karen Stewart, Senior Scientific Specialist at Technology Networks, and I'm the moderator for today's webinar. We have three fantastic presenters for today's session who will be sharing valuable insights with us. Dr. Sven Bockland is Species Director at Colossal Biosciences, a bioscience company using genome editing to restore extinct animal species, protect critically endangered species, and repopulate critical ecosystems. A human geneticist by training, he spent 10 years in technical leadership positions in genome analysis companies and developed the epigenetic clock. Anthony Mastracchi IV is a research associate at Colossal Biosciences with expertise in metrology and assay development that fuels Colossal's capacity to culture non-model organisms and assess genomic stability during editing. Dr. Jessica Hartman is a Senior Director of Product Applications at Cell Microsystems. She's dedicated to pioneering advancements in cellular and genomics workflows, particularly focusing on innovative product applications within the CellRaft technology platform. Following the presentation, we'll have a Q&A session where you can raise any questions you may have for our speakers. To submit your questions, simply type them into the box on the right-hand side of your screen and click Send. We'll do our best to address as many as possible during the time allotted. If you encounter any technical issues during the webinar, please click the chat box on the right-hand side of your screen for assistance. So now, without further ado, I'm delighted to hand over to our esteemed speakers.
Hello, my name is Sven Bockland, and I'm a Species Director at Colossal Biosciences, meaning that I lead a team that's working on bringing back currently extinct animal species. We call ourselves the De-Extinction Company, and while the technology to truly create entire extinct genomes doesn't exist yet, we're making great progress in identifying large numbers of genes and gene variants that have gone extinct with these extinct animals, and we are bringing those back to de-extinct these traits and to make proxy species of animals with those extinct traits right now. And for the next few minutes, I want to explain why we do what we do. We're in the middle of the sixth mass extinction, similar in scale and speed on a geological scale to the one that wiped out the dinosaurs. We are watching an immense loss in biodiversity, and besides all these numbers shown here of animals that are on the brink of extinction, there's some more stunning statistics. So of all the animals on Earth, 96% of the biomass are humans and the livestock that we grow to feed us. So only 4% is left of animals that is wildlife. There are more chickens on this planet than all the other birds of all species combined by far. And that is terrifying. And of course, that is caused by habitat loss caused by our expansion. So this mass extinction is like the asteroid, except we are the asteroid. And we can choose to watch, or we can try to stop and reverse this. And our very ambitious goal at Colossal is to make extinction a thing of the past. Now, of course, to do so, we need to stop and reverse climate change and habitat loss most of all. But our part is this. We restore extinct species to rebuild ecosystems. To do so, we're developing advanced technologies, including reproductive technologies, genetic, cellular, and molecular technologies. But then if you look on the right here, we're using these technologies now to help protect the species that are endangered and to enhance conservation programs that are in need of some updating with all these modern technologies. So far, Colossal has announced 3 extinct species that we are working to bring back. The woolly mammoth is probably the most famous of them all. It went extinct just a couple thousand years ago, and we are hoping to rewild those into the Arctic, for example, in Alaska, but the rest of the Arctic as well. The thylacine, also known as a Tasmanian tiger, was a large marsupial carnivore, a little bit like a fox or wolf in size. And it was really the apex predator of Tasmania and Australia. And when that animal went extinct, because we put a bounty on its pelt, we hunted to extinction. When that animal went extinct, it really collapsed the entire ecosystem in Tasmania. The role of predators in ecosystems are really important. And when we eliminate those as humans, there's dire consequences. And then the dodo is really the symbol of man-made extinction. It went extinct about 350 years ago, mostly by invasive species that were brought to the island of Mauritius by us, by humans. Now, besides that, we are working on these reproductive technologies, and we're testing those in a range of currently endangered species. And Anthony will talk about some of the work that we are doing with those later on. Now, overall, our genome editing workflow is something like this. We're applying this here to elephants and elephant cells, but this really works across the board. So the first step, of course, is identifying the DNA edits that we want to make. And we do that by sequencing these extinct animal genomes. In some cases, like the mammoth, we've been able to sequence more than 50 mammoth genomes. For the dodo, there's currently only one. Now, we align those to the closest living relatives, and then we have to decide which edits we want to make to get the phenotypes that we want, and then how to make those. And here's a quick list of some of the tools that we are using for this genome engineering. I'm not going to talk about all of them in detail, but we're basically using every tool that exists in the toolbox. We can use something like CRISPR and base editing to make specific, or HDR to make specific changes in the genome. We can build large genome structures in BACs and then replace them into the genome with a method, something like cast or paste, where we integrate a landing site and then use a transposase or recombinase to put that in. We can even potentially build entire artificial chromosomes that have kind of conversion genes on there for the species that we want to build, and we can do all of that with or without editing systems, reporter systems that can show us whether these edits have been made or not in the cells. Now, the hardest part really is not making these edits, but it's making them in cells that survive. You see that on the top right there. And that is really the most difficult challenge for us right now. We work with a range of immortalization methods, but the most difficult part is really that we don't work with any model organisms, or hardly any. Elephants and birds and marsupials and other of these large mammals are not exactly cell types that we can find a lot of published protocols on. So we just have to develop these protocols ourselves. You may have seen some of these news articles back in March when we announced that we made iPSCs from elephants. Elephants are really difficult cells to work with for a range of reasons. And so these iPSCs were a big deal for us. It's an important step towards our woolly mammoth work. We published this in a preprint in bioRxives, where we showed that our mammoth team needed to use a combination of chemical reprogramming, elephant-specific Yamanaka factors, and p53 repressors to create these iPSCs. But this is just one of these species, and for many of the others, the cell identification, characterization is a huge part of our work. Now, the main focus of this presentation today is on that last step here, and that is isolating these modified cells. And then, of course, we need to characterize those. We do all the editing in bulk cells, but we need monoclonal isolation to be able to stack edits into cells that we can then clone back into an animal. And we do that typically using somatic cell nuclear transfer or cloning. Now, to isolate cells to make monoclonal cell lines, the standard in the field really is to use FACS, or fluorescent automated cell sorting. Depending on the editing tools that we can use fluorescent reporters to identify the cells that we want to sort, and then deposit these one at a time into 96 well plates. Now, that puts several stresses on these cells. They sit in a sorting buffer, sometimes for a long time. They're exposed to high pressure during the sorting. And then importantly, they end up alone in a well without the ability to secrete and share growth factors with other cells. Now, we can get around that issue somewhat by using conditioned media for the first few days that these cells have grown in previously, but still our efficiency of outgrowth is quite low for many of the species that we're working with. So what we're going to talk about today is how we are using the cell raft air from cell microsystems to isolate cells in a way that is more gentle than FACS sorting. In this method here, we seed cells onto a culture plate that is made up of thousands of microscopic rafts that can pop out from the array. So the cells settle down on these rafts, but they share the media, and thus they can keep exchanging growth factors as if they are in a standard cell culture dish. By scanning these arrays regularly, we can monitor the growth and the doubling rate of the cells. We can measure fluorescence. And for each colony of cells that we want to select, we know we can go back to these images and we know whether initially that cell was alone in that raft or not, and thus whether that is a monoclonal colony or not. When we choose the colonies that we want to expand, there's a magnetic wand that will move those into a 96-well plate. As you can see here, that raft is pushed up out of the array and then moved over into its own individual well of the 96-well plate. We had some concerns at first about cells getting mixed up, but these cells really do adhere tightly to the rafts, and we've thoroughly confirmed that these are monoclonal in our hands. And then when you look at the right here, you can see the cells grow off that raft. You can see how that colony expands beautifully from the raft. So this method has a range of advantages. And again, Anthony will talk about this in a lot more detail in his section on how we are using this so far. So all in all, the tools we are building for de-extinction will help us save currently endangered animals. And we're making all of these technologies available for free for conservation. But making it through these bottlenecks, making all these methods work for these really non-model species where very little work has been done in the past is an important part of our focus right now.
Thank you, Sven, for that great presentation. And we will now hand over to Anthony for his talk.
Awesome. Well, thank you so much for the introduction. Hello, my name is Anthony, and I'm a research associate here at Colossal. Some of my responsibilities revolve around generation of mouse models and the integration of newly generated cell lines into our research pipeline. One of the key hurdles our cell team encounters stems from our exploration of cell types that remain largely uncharted, compounded by the fact that we're working with species already lacking comprehensive cell characterization. Undoubtedly, the task of isolating cells harboring desired edits poses significant challenges, particularly when confronted with the nuances of post-primary derivation cell maintenance. The cell raft technology has emerged as a pivotal tool in expediting Colossal's research endeavors, notably through its capability to automate colony picking for mouse embryonic stem cells. So just to give a little bit of background, we sometimes employ mouse models within our research to generate chimeras, and this aids in the elucidation of gene functionality and intergenic interactions. This particularly is useful for supporting our projects for non-model organisms, such as hoofstock and avian. So to anyone who's familiar with mouse embryonic stem cells can surely understand the tedious process of spending hours hunched over a stereoscope, meticulously using a P20 pipette to manually pick clones from a 6-well flask or larger. Then each clone is placed in a PCR strip tube filled with Accutase or similar dissociation solution, followed by incubation in limited batches. 1816 colonies, and this is to prevent overdigestion before returning to pick more. This is quite labor-intensive process and places a significant strain on our scientists, consequently diverting their attention from higher-level tasks. Now, upon implementing the cell raft, we immediately noticed its ability to automatically pick cones of interest at a rate of 24 to 36 clones per batch before requiring incubation. This in turn freed up our scientists to focus on other lab tasks. And the image on the right showcases a small section of the array featuring multiple cell rafts. Highlighted in green are the ES cells of interest. Although it may be difficult to discern in this image, subsequent slides will provide clearer visuals, but we see the ES cells positioned atop of a feeder layer of embryonic fibroblasts, which in turn rests on a gelatin coating, replicating typical cell culture conditions for these cells. Again, one might notice their small size, resembling dots rather than actual colonies, and this is because each represents a single ES cell isolated within the raft 4 hours post-seeding. Now, the ability to observe individual ES cells just 4 hours after seeding expands our editing workflows and instills greater confidence in our research. This enables us to explore new avenues that were previously inaccessible. So traditionally, or should I say before the cell raft, at Colossal, our approach involved seeding ES cells at low density on a 6-well plate, followed by waiting 7 to 9 days before examining under the stereoscope. Unfortunately, this method occasionally resulted in the formation of polyclonal lines. This would manifest when 2 ES cells either landed in close proximity to one another or failed to fully dissociate, appearing as a single colony days later. Consequently, screening for polyclonality during genotyping became necessary. However, with the ability to visualize single ES cells just 4 hours post-seeding, we now possess greater confidence in the monoclonal nature of colonies being generated. And as a matter of fact, since implementing the cell raft, we have yet to encounter a polyclonal ES colony. Now, the image displayed on the right are just some examples of ES cell colonies ready for picking, with the more features visible, such as the underlying meth layer. By capturing and monitoring events at such an early stage, we're now capable of exploring novel editing modalities such as mRNA, which were previously inaccessible and steers us away from needing to use plasmids for editing. And this is because when we did plasmid editing, we would have antibiotic resistance genes for either pyromycin or hygromycin to positively select the cells, which even with the uptake of a plasmid would still leave the cells sick. Additionally, there was always some concern with random plasmid integration into the genome, which would necessitate additional screenings. Lastly, it's more biologically relevant to the cells to not be constantly bombarded with high amounts of antibiotics, especially stem cells, which some literature points to decreasing their overall pluripotency. So as I previously alluded to, one of the new editing modalities for ES cells that we're able to bring on board here at Colossal is mRNA, which has a pretty short editing window, peaking at around 8 hours and tapering off over the course of 48 hours. In these images, we see pretty early ES cell transfections. The first bottom image is a pretty early colony. I believe this was taken around 24 hours mark post-seeding. And the others were taken around 48 hours since we wanted to see how long the fluorescence could be observed. And we can see the tapering off of the fluorescence beginning to fade, resulting in low SNR. This now demonstrates to us that we can start using fluorescence for positive selection for mouse ES cells, which most consider to be model organisms, and the literature supporting their culture conditions is abundant. But what about non-model organisms? Can we use some of what we've learned and apply it to an organism that has never been characterized before? This brings us to Colossal's first case study of applying the cell raft to accelerate our workflow on monoclonal generation for cell lines we really are just beginning to understand. So similar to the mouse ES cells, we sought to transfect our hoofstock lines, then use GFP positive selection to potentially isolate clones with desired edits. In this study, we identified 597 clones which had a degree of fluorescent signal, so we focused on those clones and sought to identify their viability over time. To be on the more quantitative side, we broke the viability of potential clones into different milestone markers that we would typically observe on the cell raft for wild-type cells over the course of 6 days. So we have milestone marker 1, the qualification being that the cell has flattened out by 24 hours. Milestone marker 2, the cell is starting to divide or is still alive by 48 hours. And milestone markers 3 and 4 are really just indicators that this cell is starting to form early colonies. Milestone marker 4 is that we expect it to be fully confluent on the raft by the day 6 mark. Now, this is an image of just one of the clones that we can see making through the different milestones. Again, milestone marker 1, the cell has somewhat flattened out. Then milestone marker 2, the cell is still alive and moving around its respective raft. It's also a little bit easier to see the punctate Cas9 GFP during the stage. Now, in contrast to the mouse ESC cells where we employed mRNA, here we use RNP editing modality, where the GFP is directly fused to the Cas9. So you expect the fluorescence to be centralized in the nucleus and not be ubiquitously expressed. Now, then later, milestone marker 4, it's confluent on the cell raft. Now, to determine the degree in which our positive selection was beneficial, we conducted next generation sequencing on the bulk population of cells, which yielded roughly 42% modifications, which means on average 42% of the cells had some degree of editing. Again, this is a knockout. Therefore, even if an indel did not generate a true knockout frame shift, it's still considered a modification. And then with the roughly 150 clones we isolated from the cell raft, having a GFP signal, we found 70% of them had modifications, thereby demonstrating to us that we can use transient reporters to enrich. As for outgrowth from the cell raft, our hoofstock fibroblasts were able to achieve 95% outgrowth and expand past the 96-well stage, giving us roughly 90 clones from this experiment that were edited, including the indels, and had grown past the 96-well stage. Further, since the cells were isolated at the 8 to 16 cell stage on the rafts, we were able to use traditional media and not resort to conditioned media or adding in ROCK inhibitor, which was previously needed when we isolated these cells from flow. This video is just one of many clones which were able to grow out from the cell raft onto a 96-well plate. This was taken on the IncuSite SX5. So I'm just going to play that video again. So we have the cell raft in the middle, and then we can see over time the cells growing out or kind of growing into this 96-well plate. So from here, I thought, let's try this cell raft on one of the most difficult cell lines that we have in-house. And these turned out to be pericytes. Now, pericytes are cells that sit on the walls of blood vessels, and they aid in the vessel formation and maturation of endothelial progenitor cells. What makes them so difficult to work with is they are very dependent on one another, and when we attempt to isolate them, they almost immediately senesce. Previously, with flow experiments, we attempted different kinds of conditioned media, adding in ROCK inhibitor, adding other apoptotic drugs, and the best we got was around 10% outgrowth efficiency. Immediately, without any optimization from the cell ref side, we had jumped to 25% outgrowth efficiency. And more interestingly is when I was combing through our IncuSite images, I found the reason that they were at 25% outgrowth is not because they were senescing, but rather they did not want to have to come off the rafts. So the colonies would keep growing until they enveloped the rafts, whereby contact inhibition kicked in and halted colony expansion. So in follow-up experiments, we thought that since pericytes sit on the walls of blood vessels, their environment is very elastic, and the cell raft could be providing that elastomeric support that they like so much. So we plated a 6-well plate with collagen, and then another with matrigel, which is considered a softer substrate, and found that they were more convinced to come off the rafts. But we're still working on how to get them to come off more consistently. Nonetheless, a 2.5x increase in outgrowth efficiency is still a major game changer, which we're very happy about. So in this left image, we see the cells are growing on the raft, and this is coated on the collagen plate, but they're not wanting to come off. But then when we coat with matrigel, we can see at around the 4 to 5 day mark, they're starting to want to come off and embed themselves into the 96-well plate. So just to recap, in the past year since we've acquired the cell raft, this technology has aided us to accelerate our projects by allowing us to optimize conditions within 1 to 2 experiments, whereas with flow cytometry, we would spend weeks or months attempting to dial in pressure, voltage, attempting to add in drugs to prevent apoptosis. With the cell raft, we don't necessarily have to deal with that. It enables our scientists here to work on higher level deliverables and not spend hours picking mouse ES colonies or genotyping potentially hundreds of clones to find the one with the desired edits. And again, we see that overall our cells are healthier, and we can probe into real time how transfection is affecting our cells.
Thank you, Anthony, for those great insights. We're now pleased to hand over to Jessica for the final talk.
So as Sven introduced, most of us are well aware that gene editing has become a cornerstone of almost every biological application, from pharmacological to toxicological to developmental. And if you've ever done a workflow like this, you know, as Sven introduced, that it starts from the process of getting your genetic material into your cell of interest. This can take a variety of different forms. You can use transduction or lipid-based methods, electroporation, microinjection, or even virus to get your DNA into your cell of interest. Once you've transduced your cell population, you then let that population expand, turn it into a single cell suspension, which then has to be single cell isolated using one of these types of methodologies. This could range from the simplistic, using a micropipetter to manually put cells in a well by limiting dilution, or to the more complex, using high-speed flow cytometry with a laser-based detection system or a low-pressure fluid-based system. But regardless, in any of these methods, the goal is to put a single cell in a well and allow that cell to grow to recapitulate your phenotypic population. Now, despite all of the advances in technology and all the tools that are available to us as scientists in our toolkit, this process remains relatively challenging and time-consuming. And we performed a poll of the scientific community to get a sense of why gene editing can be so challenging in the lab. And as you can see in this results poll, the number 1 and 2 answers were that it is largely inefficient and incredibly time-consuming. And this leads to other problems like too few clones. You use too many consumables, you may not even have a clonal population when you're done, and you have to use a lot of different instrumentations to do this. So I'm going to pause here for a poll question because we'd really love to know what you're using in your own labs for your gene editing workflows. So why exactly is gene editing so challenging? Well, every step that I'm showing you here in this cartoon comes with its own caveats and its own limitations. The very act of putting genetic material into a cell can cause lots of different effects on your population as a whole. Transduction as a whole is generally inefficient, meaning that you may have anywhere from 1 to 5% of your cells getting your edit of choice. These methods can confer toxicity to your cells, leading to fragile or damaged cells. And ultimately, you're left with a heterogeneous population that may not be fully edited and only be heterogeneous, or you may only have a very rare variant in your population that you're trying to recover using these sorting-based methods. Once you've transduced your cells, you take this fragile population of cells and you then use them in one of these other methodologies, which causes fluidic pressure because of the high pressure or low pressure systems that you're using or the manual pipetting. That fluidic pressure can lead to cellular damage and cell stress. And in addition, you have inefficiencies in the plating of these where you may not even have a single cell in a well, or if you have a single cell in a well, that cell has already gone through a lot of stress to get where it is. That single cell in a well now has the hurdle to overcome to recapitulate the population. And because that cell is alone, you have that single stress cell, there's no cellular crosstalk, and more often than not, that leads to a single dead cell in a well and not a happy, healthy, genetically modified population that you can use in your downstream research. So at Cell Microsystems, the core question that we challenge our customers with is, what if you didn't have to put a single cell in a well? And as Sven has introduced, that's really the main reason our technology is so much more efficient at gene editing than any other technology. Using our proprietary dish, the cell raft array, we're able to seed tens of thousands of cells in a single small volume. When we seed those cells, they settle into tens of thousands of individual microwells, which allows us to culture the cells in a favorable environment while also maintaining spatial segregation of the cells. When you go into the lab with the cell raft array, it's really as simple as taking that cell suspension that you're used to generating and manually pipetting it into the array. There's no fluidics, there's no pressure, you just simply droplet dispense into the well. As the cells settle into the microwells, as you can see here in this cartoon, they float by gravity into the microwells where they will attach onto that cell raft, which you see in cartoon in gold. As they attach, they will adhere. And while there are tens of thousands of rafts, some may be empty, some may have multiple cells, really what we care about is that cell in the middle, the rafts that contain a single cell of interest. We can put that array into a standard tissue culture incubator, culture the cells over days or weeks in order to grow out the population that we actually care about. And as Sven has already highlighted, when we find that population, we're able to retrieve that cell raft and use it for downstream expansion. Now, I've shown you a cartoon of one raft, but really the great thing about the array is that we are able to do this on a frankly unprecedented scale. What you're seeing here is a stitched together complete field of view image of every single cell raft in the array. And each of those red rafts represents a pluripotent iPSC colony, for example, that we could recover for downstream propagation. So in that one small 3 ml volume on the left-hand side, we can replace literally dozens of traditional consumables, either 96 well plates or 384 plates, saving you time, energy, reagents, and really getting you to your downstream endpoints far faster than you would in a traditional workflow. Now, as we all know, thousands of wells sounds really wonderful in theory, but in practice, very few biologists have the patience or the inclination to look manually through every single cell raft. And that is where our software analysis really helps you identify exactly the cell rafts that you're looking for your downstream workflows. We call this image-based software-guided cell raft selection, and we can use the images that we take of the cell raft array with our software to refine the cell rafts that contain our clones of interest. We can use this software to identify cell rafts containing single cells at time points, for example, time point 1. And we can then identify the cells of interest based off of morphometric characteristics such as size or shape. Using the images over time, we can monitor those clones for confluency. And if we're lucky enough to have a fluorescent marker, we can use fluorescence-based imaging to determine if the cells express our marker of interest. Using cytometry, we're able to triage those thousands of rafts down to a curated list containing only the cell rafts that we care about. And then we can very easily map those cell rafts for isolation and downstream recovery. So when I say isolation, we consider this to be intelligent isolation using our instrument, the cell raft air. The air is what you see here on the left-hand side, and it's pretty much what you see is what you get. It's completely fluidics free. It's very simple to use. There's no sheath fluid, no tubing, absolutely no pressure. The isolation of our cell rafts is fully automated. It's very gentle, and we're isolating the rafts themselves, not single cells. That means there's no trypsinization, no scraping, no aspiration, no pruning, no manual dissection. It happens fully hands-free just by that automated transfer. What that means is that when we do this isolation process, we're filling your 96 well plates with established pre-selected colonies that meet your criteria, not single cells that have undergone a lot of selective pressure and stress. So after your cells have had time to grow on the cell raft array, we're able to use the software settings that we've generated to map the rafts for isolation and recovery out of the proprietary consumable. When we retrieve them, we take that cell raft that has our colony of interest, we dislodge it from the array, and the raft gets transferred over to a 96 well collection plate for propagation. The raft falls passively to the floor of the 96 well plate, and then it will grow off the raft, just like you see here in the cartoon, to colonize the floor of the 96 well plate. You can see here a bright field image of cells growing off of the cell raft after transfer. You can see at day 1, the cells are on the raft, and they just will walk off passively over time. The raft will eventually go away with regular media changes, and once the colony has reached a substantial size, you can passage that colony through any normal methodology that you're normally using, and it's ready to go into any other tissue culture dish for your downstream workflows. Now, the best part about the technology is that while we have transferred a colony of interest, we are able to understand the entire track and trace lineage of that clone from start to finish. What you're looking at here in the bright field images is a comparative workflow in a gene editing experiment using CRISPR editing in A549 cells after 11 days post-transduction. In the top bright field image, you can see the cell raft in the center, a very large and beautiful homogeneous clone that's grown off the raft that was ready to split. And you can see happy cells that are ready for expansion only 12 days post-transduction. In comparison, in one of the lone wells containing a colony after limiting dilution, you can see a very small colony with heterogeneous looking cells. And upon passage, that colony did not survive. But importantly, we can track back for our cell raft air colony to the original cell that generated that clone. So as you can see here in the images from the instrument, at day 0, we have a very beautiful single cell. That cell grows into a clone of interest and of course generates that colony that you see here on the right-hand side. In contrast, if anybody has ever tried to find a single cell in the well of a 96 or a 384 well plate, you know that's basically impossible. And so we have no ability to confirm monoclonality in those limiting dilution-based workflows like we do with the cell raft air system. So what that means is when you use the cell raft array, you're able to easily identify clones, transfer them, get your colony of interest, and then have assurance at the end of that process that you already have a colony and you don't have to spend as much time on the downstream orthogonally confirming that you do in fact have a clone. So as I already alluded to, we're not just limited to the bright field imaging, we're also able to use fluorescent staining on the array in order to help us find the cells or colonies of interest. To me, I think this is one of the most amazing tools in our toolkit with the air system, because if you've ever performed immunofluorescence or flow-based sorting, you know that the real power is in the combination of those two things. With immunofluorescence, you're able to get beautiful pictures of adhered cells that you can look at, but you can't actually recover those cells from the dish on an individual level. With flow-based sorting, you can stain your cells and recover them, but you don't get an image of what the actual morphology or the staining expression looks like in terms of the patterning on your cell of interest. With the air system, we can actually combine those two powers to antibody stain our cells in situ, image them on the array, and then recover our antibody stained or our imaged cells for further expansion from the live cells. So what you're looking at here on the left-hand side is a population of suspension cells that were stained for a surface marker. You can see expressed here in the GFP, that expression in the array as a field of view, as well as on a single raft level, and we're able to identify positively stained cells and recover them for downstream expansion. On the right-hand side, you're looking at pluripotent, induced pluripotent stem cell colonies that we are staining with a pluripotency marker, TRA160 in the red. We can use that red to help us identify clones that have not early differentiated and actually recover pluripotent clones for downstream propagation. So with the combination of all these tools, we like to say that you can say goodbye to sorting and hello to rafting, because we can use the combination of these technologies to speed up our cell line development workflows. So with the array itself, we enable this flask-like culture conditions of tens of thousands of cells while also letting us do single cell separation. We can use both bright field and fluorescence imaging with our software to identify the clones of interest. And then we're going to transfer those target colonies over for automated retrieval on the air system, enabling us to get to the colonies that we care about much faster. So why are we so much more efficient with cell raft technology than other single cell technologies? For one, because of that flask environment and that shared culture media, we see a superior viability of even the most difficult to clone single cells on the array. Because the cells are single, but they're not actually alone, we see that virtually all of the single cells will actually be able to expand and grow on the array, allowing us to get clonal colonies much faster. What that also means is that we have the ability to select high quality targets for isolation. In the vast majority of these workflows, you are faced with a survival of the fittest type situation where you may have 3 or 4 clones surviving and you are forced to use those as your clones because that's all you have. Because of all of the colonies that we have available to us on the cell raft array, we aren't limited to just the survivors. We can pick the highest quality cells that we have the highest confidence in for cell raft selection. We are going to transfer those cell rafts that you care about using completely perturbation-free outgrowth, which ultimately preserves viability and your phenotype. So what we see is that the clones that we transfer are homogeneous and they're stable, leading to maintenance of your phenotype over time. This gives you in the lab an overall improved likelihood of achieving experimental success because you have stacked the deck in your favor and you are letting the biology work for you and not against you. And what I think is really important to emphasize is that we are able to enable cell line development, as Sven has already alluded to, from even the most challenging cell types. Most everybody has successfully cloned a 293T or a CHO cell line. It's much harder to do this with cells that don't behave quite as favorably as those types of immortalized cell lines. So we've done this from a whole host of animal types and species types, and you're going to hear more about this from Anthony in a moment. So just to highlight some of the work that we have done to solve these challenging problems in the field, we performed what we call the Cell Microsystems Clone Challenge. In the Clone Challenge, we invited the scientific community as a whole to submit projects that they had been executing that had failed traditional methods of cell line development, whether it's limiting dilution or high-speed flow cytometry. And then we committed to using CellRef technology to solving these problems in the real world. So to show you what some of those problems were, each of these investigators had what I would consider to be a very common problem in cell line development that had stymied their abilities to move their scientific progress forward. In this first example, we were working with a researcher that was growing acute myelogenous leukemia cells. The cells had poor viability as well as incomplete knockout of their gene after CRISPR editing. We worked with a researcher who was trying to derive multiple chemo-resistant clonal cell lines to multiple chemo drugs in the lab. They'd been working on this project for over a year and had failed to derive a single stable cell line due to the chemo resistance drifting over time. We worked with a group using a glioblastoma cell line that has a rare genetic variant in the heterogeneous population. Their goal was to derive isogenic cell lines of the two variants in the population, and the rare variant represented only a 1 in 5 chance within the population. They'd been attempting to derive monoclonal cells using traditional methodologies, but these cells grow very slowly and are prone to premature differentiation, so they were unable to derive isogenic pairs. In a similar problem with the drifting of the phenotypic response, we were working with a group that was looking at hypoxic responses. They were able to derive clones by limiting dilution. However, every clone that survived limiting dilution had lost its ability to respond properly to hypoxia, both from a phenotypic and a transcriptomic perspective. This next researcher was trying to derive a stable reporter cell line from a patient-derived induced pluripotent cell line. These iPSCs were very fragile after flow-based methods. They had tried both flow cytometry as well as droplet dispensing using a low-pressure sorter. The iPSCs failed to survive after sorting, and in addition, their fluorescent reporter was not expressed until after differentiation, so they were unable to use fluorescent labeling for identifying potential clones of interest. And lastly, also using fluorescence, we worked with a researcher who was attempting to derive a double positive reporter cell line to look at drug sensitivity in a cancer project. These two reporters were independent, meaning that each of the lines that they derived had varying fluorescent intensities, and they were unable to derive a stable double positive with high fluorescent intensity for both of their markers. So each of these clone challenges sent their cells to our lab here in North Carolina, and we were able to work with each of these researchers to try to solve these problems and derive stable lines that they could use for their research. So what I'm going to show you next are the actual data from the investigators after we transferred clones back to each of the labs, and they were able to use orthogonal methods to test the clones that we provided to them. So in the first example with poor viability and incomplete knockout, we were able to provide the researcher over 30 clones that they were able to use to confirm the edit as well as the clonality of the population. For the chemo-resistant population, we were able to derive dozens of clonally resistant cell lines that they confirmed were in fact still chemo-resistant after transfer back to them, and we were able to do it for 2 different cell lines for both of the drugs that the researcher was studying. For the rare genetic variant in the heterogeneous population, we sent 30 different clones, and they were able to derive that isogenic pair that they so desperately wanted, which they were able to confirm each of the variants by doing FISH, as you can see here in the FISH image on the right-hand side. For the researchers looking for hypoxic responses, we were able to derive 6 different clones that maintained a vastly improved hypoxic response compared to the traditional response that they saw in the heterogeneous population that they could then use in their downstream workflows. The iPSC reporter cell line, we were in fact able to derive a monoclonal edited iPSC cell line. We then differentiated that iPSC line into mature neurons. And what you see here is the expression of that reporter gene in green, as well as mature neuronal markers, proving that the cells maintain their ability to differentiate as well as turn on the reporter of interest. And lastly, we were able to derive that double positive clonal reporter cell line that gave the researcher high fluorescence intensity for both of his genes of interest. So we were really pleased to be able to solve all 6 of the clone challenges that we embarked upon. And this really highlights the breadth of what the AIR system is capable of and how it can really accelerate and solve challenges that you're having in the lab. And I want to thank our collaborators specifically in the clone challenge, Johns Hopkins, UNC, Stanford, Columbia, NC State, and Georgia State, all of whom helped us embark on these exciting projects and can now use these clones in their own labs. So if you have a challenge that you want to give to us, we'd love to help you too. We are embarking on the clone challenge again, and we would love to hear what your research involves. If you want to submit, you can apply at our website down below. Submit a short abstract about the challenges that you're having doing celling development. We'll select 5 winners just like before, and those applicants will work with our team here to develop the protocols, and then we will verify single cell derived clones and send them back to you. So I'm going to pause here for a poll question to hear about your interest in CellRAF technology.
Thank you all very much. I'm sure our audience will agree that the presentations have provided some really valuable insights and raised some excellent points. So just to make you aware, there will be a survey at the end of the webinar, and we'd very much appreciate your participation. But first, we're going to move on to the Q&A session. To pose your question to any of our speakers, please use the box on the right-hand side of the screen and click send, and we will do our best to answer as many questions as possible with the time available. So we've already received a few, so I'm going to dive into the first one. How many clones can I expect to get with the CellRAF instrument?
That's a great question. It really depends a little bit on what your cell type is and how many cells that you've seeded. But in general, we find that most researchers are very easily able to fill several 96-well plates. And it's almost more dependent on your desire to screen than it is the number of clones. So we find that you're very rarely limited to the number of clones that you'll have available to you. But for the most part, I would say several hundred on an average basis.
Excellent. Thank you very much for that response. And our next question. For ES cell optimizations, how did your team determine optimal conditions?
Ooh, that's a good question. So to determine ESL optimization, we knew that the cell rafts are made of the same plastic that our cell culture flasks are made of. So we knew that we could coat with gelatin and then MEFs on top of it. So what we first did with the MEFs was empirically determine the ratio of mouse embryonic fibroblasts to rafts that we would need in order to actually cover the raft in its entirety with MEFs. So we ended up using a quad array, and we did a 50 to 1, 75 to 1, 100 to 1, and then a 200 to 1. And then from there, we determined over the course of about 2 days, when those MEFs kind of layered out into the rafts, what seeding density was most optimal for those. And then when it came to seeding the ES cells, we again used the quad array, and we tried different seeding densities to determine what was the lowest density that we could go with to maximize the amount of monoclonals that we would output, but also not too low that the embryonic stem cells no longer had like those paracrine factors in their environment.
Excellent. Thank you very much. And our next question. Can the cell raft air find my rare event or low efficiency edit?
Yes, that's actually what we excel at. When you're trying to find a rare or an unusual edit, a lot of times you're limited by, as I mentioned before, what survives from your 96-well plate. So we've had a lot of success stories from our customers who've told us that because of the sheer volume of clones and the health and the viability of them, they're able to identify much more challenging and rare edits, particularly if you have a fluorochrome, as we can use the fluorescence to identify those rare cells and then recover them with a high viability and high success rate. The other key advantage is that we are able to enable most of those cells to grow, even as single cells on the array. And what we find is that even if you have a phenotype that is affected by your gene edit, for example, your cells slow down, growth rate rise, or the phenotype is different, we can track that using the software. And that allows us to also improve the editing efficiency and the recovery of even the rarest of the edits.
Excellent. Thank you very much. Next question. In the hoofstock slide, how was the viability of the cells determined?
Yes, so for our hoofstock, since they were fibroblast, what we determined was that if the cells were dying off on the raft, then there would be essentially two fates. The first fate was that they would start to ball up. And so you would see that circularity kind of increase in the cell. And then the texture would also go up. So they would start to look more of like raisins. And so both of those are features that the Cell Raft Air software can look for. And so it was more of just setting that gating to say, hey, if the fibroblasts had started growing out, but then we see circularity start to increase and it's starting to become ruffled up, then we knew that those were starting to either senesce or die off.
Excellent. Thank you very much. This one's directed to Colossal. The Yangtze giant softshell turtle, the world's largest freshwater turtle, is now critically endangered, with only 2 known males remaining. The last known female, discovered in Vietnam, recently washed up dead. If she had survived, she could have laid hundreds of eggs each year. Unfortunately, the species is now essentially doomed to extinction. One suspects that the female's tissue has been preserved. Is bringing the Yangtze giant softshell turtle back from the brink something that would be in your mission? A potential related species that could be used in hybridization is the Chinese softshell turtle.
Yeah, I think this is a clear example of a species where Colossal's technology could really help. It's a very similar story to that of the northern white rhino that is functionally extinct now, with only 2 females remaining that are too old to breed. And so we're working actively with the rest of the scientific community in helping bring back that species. Now, turtles, of course, are a whole different kind of animal than the mammals that we're working with. And so I think in this case, we'd probably wait to see how much progress we make on our dodo project, because of course there also the gene editing will happen in eggs, as we would probably do with this turtle. So this is something that I think is a great example of a species we would love to work on. My guess is that we need to make some progress on the dodo first before we can move on to that.
Excellent. Thank you very much. We'll definitely be watching this space. Next question. How have you assessed the potential impacts of reintroducing those extinct species in ecosystems that have evolved since their extinction?
Yeah, what you have to keep in mind is that reintroducing any of the species we will bring back is not going to be an overnight project. It's going to take years or decades to build large breedable populations, and so reintroduction will happen step by step. The most logical step is to first have the animals live in a really controlled environment, say in a state park or a national park, where we know exactly where the animals are, what they're doing, how they're interacting, and we can control the other species that interact with them. And then as that data comes in, I think we can plan on wider introductions as well. But already we're doing some research. Just I think a week or 2 ago, we published a paper that calculated the density that mammoths could probably live in the Arctic. And so we're working with the scientific community, with academic labs around the world, and with, most importantly, conservation organizations to make sure that any potential reintroduction will happen in a way that benefits the animals and the ecosystem.
Excellent. Thank you very much for that. On to our next question. Does the raft work for suspension cells?
Yes, that's a great question. It actually does. The key is ensuring that these suspension cells can be adhered to the raft. Of course, with the raft being the transport vessel to recover the colonies, you have to adhere the suspension cells to the rafts themselves. We achieve this through a variety of methods that are cell line dependent that we can help with and that we've optimized and have protocols for. It's typically a very transient attachment to the raft, and then after transfer, the suspension cells will go back into suspension in the 96 well plate, and then they can be expanded off raft just like they would be in normal suspension cell culture.
Excellent, thank you very much. And our next question. Do we also need to make sure that there are greater protections for species that might be brought back to ensure that, like the thylacine, they're not hunted to re-extinction? I realise we're quite far down the line.
Yeah, I think I covered some of that in my previous answer, in that reintegration will be slowly and controlled, and initially in a protected environment. Now, we are already working on these kind of topics. For example, for the thylacine, we are currently working with rewilding organizations, with conservation organizations in Tasmania, with the Tasmanian government, and even with companies like logging companies, because those are the ones that determine whether that ecosystem where the Tasmanian tiger will survive will even still be there, and who will be most impacted by this. So we are actively working on that, and we will make sure that these protections are in place.
Brilliant. Thank you very much. So unfortunately, that's all we have time for today. A massive thank you to all of our speakers, and rest assured that any questions we didn't have time to get to, you will be contacted offline with an answer as soon as possible. And you can continue to ask questions even if you're watching this webinar on demand. Remember, you can download your certificate of attendance from the handouts tab on the right-hand side of the webinar platform. And thank you everyone for your time and participation. Please don't forget to complete the survey at the end that will be appearing shortly. Thank you all again.